Hey, Aaron's Bio folks. Just wanted to do a quick little session here on DNA. So if you haven't, download the Intro to DNA skeletal notes so you can follow along. But basically, I wanted to walk you through the basic rules of bonding for making structures called DNA, right? So what is DNA? DNA, the D is deoxyribo, the N is nucleic, the A is acid. So deoxyribonucleic acid is the structure that is built by nucleotides to make a piece of DNA. So when you look at it, and I think we know this one, but there are three parts that make up a nucleotide. Nucleotides chain together to make DNA. So what is the part shown in blue here on the diagram? Right here and right here. This is the simplified drawing. This is the more complex chemical drawing. But the blue feature is a sugar called deoxyribose, right? So we can put that one in there. It is a sugar that has 5 carbons. Glucose has 6, so don't be confused by it. But that is the main component of the part of it is the sugar. In yellow, so right here and right kind of an orangish yellow there, we have our second piece, which is the phosphate group. And then in green, right here and right here, we have what we call one of 4 nitrogen bases. So those 3 pieces put together makes a nucleotide. Nucleotides chain together to make actual DNA. All right, so we got the parts. So what do we do from there? Well, the structure of DNA, you take the phosphate of 1, as we said, which is I'll do it in orange. And then the blue was the sugar. So the way the sugar fits is here. So there's my sugar. Now there is a branch out like that. So if you were to count carbons, remember we said this is a 6 carbon. So there actually is a unique special way to do it. You start at right after the oxygen and you go clockwise. So this would be carbon number 1. Let's do it in black so you can see it. Carbon number 2, carbon number 3, carbon number 4, and then remember ends and bends, carbon number 5, and then this one would bond to that phosphate. And then in green, coming off of this, we would have the 1 of 4, and it doesn't, sorry, turning green here, we have our nitrogen base, and it could be 1 of 4 of those. So that is your nucleotide. The type of bond that builds a nucleotide is a covalent bond. So that sharing of electrons that we learned back in our chemistry unit. So far, that's it. That is one nucleotide. Now, when you look at DNA, this is a piece of DNA. This is half of a piece of DNA. So what you have, and I'm going to use my highlighter here to kind of show you what's what. But we have in blue our sugar. So there is one right there. There's another one. And there is another one. So the blue is the sugar, right? So I'll write that in here in a second in a key. The phosphate is this portion right here. So there's 1, there's 2, and there's 3. So that's the phosphate. And then the nitrogen bases in green are this portion right here, and here, and here. So what we have so far is a half of a piece of DNA. So the sugar was here, the phosphate was here, and the nitrogen base was here. So if you were to look at it, this is one nucleotide. Here's another one. And here is the third one. All right, so there are 3 nucleotides attached right now. That's the basic structure of DNA. But if this was it, DNA right now would have the backbone and then half the ladder, but we're missing the other half. So we have to figure out how to attach the other side. Now, you guys learned in the team activity there were some basic bonding rules for these things. So we have 4 of these bases. We have one called cytosine, which we label with a C. We have guanine, that's the G. We have adenine, that's the A. And we have thymine, and that's the T. So there is something special about these, right? There are two types of bases. The first type are called pyrimidines. The second are called, let's use pink, purines. So of the four of them, two of them are pyrimidines and two of them are purines. Now, I said this in class, but the way that I remember is that pyrimidines have the letter Y in them. So cytosine is a pyrimidine because the letter Y is there, and so does thymine. Right? What makes them a pyrimidine? Well, the thing about them is they are a single ring structure, right? That's it, which means in that ring they have two nitrogens, right? And you can see that. That's what makes a pyrimidine. What makes a purine? They are a double ring structure, and in the rings they have four nitrogens, right? Just in the rings. So which ones are the purines? Well, if they're not those two, then it has to be the others. So G and A are the purines. Right? So that is something that we've already kind of established, right? We know that there are these four bases. Well, two of them are pyrimidines, two of them are purines, and I will show you this one right here. This is going to be thymine. You do not have to remember exactly which one is which. This one is cytosine. Although you will be able to figure it out. This one is adenine, and this one is guanine. All right, so we know that. We also know, bonding-wise, that T's are going to bond with A's and G's with C's, and I'll show you why here in just a second. But we've got the bases. So when we put them together, right, we have that big arrow is pointing to the part that puts them together. Here we pull in our good old hydrogen bonds, right? A slightly weaker bond so that it's easily broken because you're going to constantly be breaking these open and closed. Right? But this is showing you between this nucleotide of cytosine and this nucleotide of guanine, you can see we have 1, 2, 3 H bonds. So the structure of the nitrogen base is what determines which ones bond with which. So cytosine and guanine have 3. So if we look at our bonding rules, right, and we just sort of clarified this, on the top right here, which two are being bonded? Well, I know this one is a pyrimidine and this one is a purine, right? So, but which one is which? Well, I see that there are two bonds between them. So I know there's one of them that bonds with 2 H bonds and one of them that bonds with 3 H bonds. Which is which? Well, A binds with T with 2 bonds, and C binds with G with 3 bonds. So my question to you is, which one is this? There's 2 bonds, so it's either an A or a T. So how do I figure out which one this one is? Well, it is a single ring structure, right? So it's a pyrimidine. Of these two, which one is the pyrimidine? Thymine has the Y in it, so I can figure out which one is which. So if that's thymine, then this one has to be adenine. This would be a good higher level test question because you got to know a bunch of stuff to figure these ones out. For the bottom one, right, I see 3 bonds, so I know it's G and C. And again, this one is the pyrimidine, single ring. So between C and G, which is the pyrimidine? Yep, cytosine. So it's a good way to put a bunch of stuff together to figure out what is what. So I now know the bonding rules. So the final structure then, if you look, and this is what you guys built in class, here's your sugar, here's your sugar, here's your phosphate, here's your base. Sugar, phosphate, base. Sugar, phosphate, base. So we're stringing together nucleotides. There's my G, 3 bonds to a C. There's my T, 2 bonds to an A. we get the double helix. Now, is there anything we haven't talked about? Well, there's one aspect that can get confusing, and it's how do I read which direction is which? This doesn't address it. It does right here. But how do I address how to read it? Remember, I counted those carbons, 1, 2, 3, 4, 5. So we have what is called the 3' and the 5' ends. DNA is always, always, always read from 3' to 5'. So in this drawing, because the 3' sugar, and again, it's 1, 2, 3 carbon, there's nothing there, it's empty. That is the 3' end. Go up to the top one. 1, 2, 3, 4, 5. The 5th carbon, it is not empty. There's a phosphate. That labels the 5' end. So this one is going to be read in that direction. Now, the other side is anti-parallel. It is going to be read in this direction. All right, so this is the 3'. Now, technically, they didn't show it, right? But there is a sugar here, which we can't see. But there is, it basically stops. And then on this side would be the 5' because it has a phosphate coming out the end. So anti-parallel, 3 to 5. They're going to read opposite. So again, you guys built this in class. You have a nice picture of yours. But when you put it all together, you have in your nucleus, you have DNA. That DNA is made of chromosomes. Chromosomes are made up of DNA, and here is what we are learning. Now, there are parts to it. So this DNA, I mentioned in a couple of my classes, if you took all the DNA out of all your bodies, lined it up end to end, out of a single person, this is one person, all the cells, it would go to the moon and back about 450 times. It's crazy. How does it all fit in there? Well, it's all folded and coiled up around specific things. So this DNA that you're learning has to then be like super coiled around itself and folded like crazy around these things called histones. Histones are a protein that allows the DNA to pack itself in there. It lets it wrap around it. Those then wrap around another coiled thing to make nucleosomes, which are what chromosomes are. And every cell of yours has 46 of these in it. Every single cell. It's crazy. Anyway, just wanted to highlight a few things about the DNA. The hardest part about it is making sure you know what the 3' and the 5' end is. That is the key. If you read DNA backwards, it is a completely different, wrong message, and that is not what we want to do. There you go. Thanks for watching. We'll see you in class.